So my name's Tony Pridmore from the University of Birmingham, and I'm going to try to make this go as smoothly as Rick would have done, at least nearly as smooth as Rick would have done. Today's webinar is by Helge Aasen from ETH in Zurich, and I'll introduce Helge in a moment. First, a little tiny bit about the working group, people who used to us. Yeah. We are a working group of the International Plant Phenotyping Network. You can see, hopefully, the people on the screen who are primarily involved in it. Myself, Anochar, Sata Sataris, Rick, and Catlin from Sydney, Australia. And we basically try to organize events, webinars, workshops, and so on in the general area of imaging and image analysis of phenotyping. You might see things like the computer vision plant phenotyping, computer problems in plant phenotyping workshop, and things like that. But these webinars are becoming quite a feature of the working group and have been very well received. Okay. We have a website hanging off the main IPPN website. If you want to join us, find out a bit more. You can also there see the recordings of all the previous webinars that we've run, and this one will be added to it later. So today's webinar is by Helge Aasen. This is going to be on quantitative remote sensing at ultra-high resolution with UAV spectroscopy. Helge is a researcher at ETH Zurich, where he leads the PhenoFly group, and I'll let him tell you everything else you need to know about him and about his work. I have muted everyone's microphones so that you should only be able to hear Helge. There's a chat box as part of Join Me. If you want to ask questions, please put your questions in the chat box, and we'll ask some of Helge at the end of the webinar. Okay.
Thanks a lot. The first question from my side is, can you hear me?
So I can hear you, yes. I've muted everyone else's microphones, so they won't be able to tell you unless they tell you in the chat box.
Yes, and I see at the chat box there are a lot of yeses, so that's fine. I still heard the beeps as well, so I don't know where they come from.
Me. I have, every time someone joins the thing, my machine beeps. I will mute myself in a second.
Okay, perfect. But then, yeah, then last question, do you see my slides?
Yes.
Perfect. So then I think we are settled, huh?
Mm-hmm.
Great. So then I guess I can start.
Yeah.
Okay. Yeah. So Tony, thanks a lot for the introduction, and thanks a lot everyone for joining this webinar. It's a great honor for me to be invited here, and I'm very happy to be able to speak here. So I will talk a bit about low altitude, high resolution remote sensing for quantitative field phenotyping. So I will not just talk about imaging spectroscopy, but also about other remote sensing methods, namely RGB, UAV imaging for field phenotyping. And the stuff I'm presenting today is not just something I have done on my own. It's a lot which have, like, a lot of people have contributed to it. So first and foremost, it's our group here, the crop science group at ETH in Zurich, with the PhD students here in the group, but also, of course, the whole working group which has supported this. Then also people from a former group at the University of Cologne in the remote sensing and GIS group there, mainly for the content of this talk. It was mainly Andreas Bolten and Georg Baret, and also some other researchers from around the world, which has, together with me, assembled a review paper recently on quantitative UAV spectroscopy. And this was Eija Honkavara from the Geospatial Institute in Finland, Arco Lecher from the Terra Luma group in Australia, and Pablo Zacco Tejada from formerly the Joint Research Center of the European Commission in Italy. So thanks a lot to all these people. And yeah, with this, I would like to start with my talk. So, well, I guess we are all here because what we want to do is to learn more about our environment, and specifically the crops which are growing in our environment. And then if you talk about field phenotyping setting, then we have our experimental plots, for instance, which are in the environment, and we want to kind of sense these and represent them in a way in a digital representation, because in this digital representation we can use this to kind of ask questions on how our crops are growing and what is happening there. But first we need to do this to transform particles or objects in the environment. So for instance, a chlorophyll molecule or the leaves or green cover of our crops, and then represent this into the digital representation. And the question is, how do we do this? And well, the topic of today is using UAVs to exactly do this and generate this data product, which then tells us more about our environment and the crops, how they grow in the environment. But the whole thing is not so easy, because already in the environment, our signal, which we want to sense, is influenced, for instance, from different illumination conditions, or from wind, which is blowing across the field and is moving our crops. But then also our sensing system influences the data which we capture. So,
excuse me, Helge, sorry to interrupt you. Could you possibly change the screen that you're sharing?
I see that there is some people who say I share the wrong screen. So
yes,
I will
we can see your preview.
Wait a second and see if I can do it differently.
We can see what you have.
So you said
slide and the next one.
Let's see. We can adapt. So okay. The people in the chat, can you confirm that you now see my slides in full screen? Okay, perfect. So sorry for this. Okay, so now you can see the full screen now. Perfect. Okay, sorry for that, but let's continue. Where were we? Okay, so we want to sense what we have in our environment, but this is then also influenced by our sensing system. So, for instance, from the camera systems we use, while the signal is transferred through the lens system, it is influenced also how it is recorded on a chip. This also influences the signal. And then in the next step, we process our data in some way. So during this processing, we also influence our data by the kind of choices we make for different parameters, how we process the data. And then, well, last but not least, then we save it in some way in a data product, but then also this is influencing our data. So for instance, if we save it as 16-bit, this will give us a different representation than if we save it at 8-bit data depth. And what is really important here is that we keep track on this whole process, and that we understand what is happening, and that we can then use this information, how we have influenced our data, to then really make sense of the data. And this is done by metadata, which then transforms the data into real information. And what we in the end want to have is information. So here at ETH, I'm the team leader of the team PhenoFly, and I just put up here our mission statement. So in the PhenoFly teams develop sensing systems and analysis procedures that deliver high quantitative data to capture reliable information about vegetation. And our vision is that we free high throughput phenotyping from large-scale facilities to understand the interaction of plants in their natural environments, and ultimately to facilitate a more sustainable use of resources. Because if we have facilities that can screen our crops in a very nice high resolution. That's very nice. But we need to do it in multiple environments. And that's where we come into play, because then we need procedures that can be applied more or less everywhere in all our field traits. And to do this, we also go through a whole chain of the sensing procedure. So we start with thinking about flight planning, setup, and flight. Then we think about data processing. And then in the end, of course, we also think about trait extraction. And I think it's very important to do all of this together, because just if you understand the full sensing process, then you will really understand what you have in the data. And this is really crucial, because if you extract the traits from the data, then you need to know what is in the data to really then kind of make sense of these traits. And then, of course, we also, then if you have extracted the traits, we put them into a database. And also this is something we work on. And then we kind of ultimately try to solve, let's say, or to approach this phenotype by equals genotype by environment equitation. So what I would like to do today is to go through this process and show you some examples of our work here, where I think maybe these are also useful for your work, so you can kind of learn from it, or you can discuss this and see, and then improve and continue on your work. So the first thing I will do is I will take an example of flight planning, setup, and the flight. Then I will go to the data processing and have an example there. And last but not least, I will also go to a novel way for plant trait extraction. Okay, so let's start. So mission planning. In my opinion, already this step is very crucial, and I think also that this should be done even before you start buying new equipment. Because depending on what you want to sense, you need to think of what equipment is suited to do this. So you need to think of the selection of the right equipment. You need to think of flight planning for the individual missions, but also what you in the end need. So what platform is suitable for my application. And then, of course, you also need to think about legislation, weather, security, and health measurements or measures. But like the last point, I will not cover in my talk today. But this already, the mission planning and flight planning can be very complex, because you need to think about a lot of things. So you need to think of what data product do I want? Do I need a point cloud to extract the traits that I want? Is a digital surface model the right thing, or an autumn photo? Then also you need to think of sensors. So do I need a point, line, or 2D imager? What data type do I need? Is it enough if I have RGB information, or do I really need spectral information or thermal information? Then also the coverage. So what area do you need to cover? How long can my platform fly? And so on. And then, of course, also need to think about the ground sampling distance. What ground sampling distance do I need to extract the trait I want to have? But this also, of course, then interacts again with the coverage. So these are some trade-offs you need to think of. Then also need to think of the focus distance and the depth of view of your system. And then also you need to think about things like GCP placement to really understand how or to get the right georeferencing if you have multiple flights and you want to combine them, for instance, or multi-temporal flights. And as I said before, all of these kind of interact with each other, and it's very complex to kind of grasp them all. So what I would like to do now in the first part is to touch on some of these aspects and also show you our solution, how we have kind of approached this. So the first thing you need to think of is the ground sampling distance. And I think this is quite intuitive. So depending on your ground sampling distance, your pixel size will be larger or smaller, and you will see different objects. So this is shown down here in the figure you can see down there. And what you can clearly see, if you go here on the left, you have a ground sampling distance of about 2 millimeters. And here on the right, you have a ground sampling distance of about 1 centimeter. And this is a wheat experiment. And what you can clearly see is that when the ground sampling distance increases, you will not be able to see leaves anymore, for instance. And this is a crucial thing, because if you want to do segmentation of leaves or segmentations of green cover, then yeah, you need to see what is soil and what is leaf. And then if your ground sampling distance is not high enough, then you would just have kind of blurred pixels, and you will not be able to use your extraction methods to get this information which you want to have. A second thing, which is kind of doing the same in the end with your data, is motion blur. So if you fly faster and your camera shutter speed is not fast enough, then you will get blurred pixels. And this also you can see down here. So on the first image, we have a flying speed about 4 meters per second. And in the last image, we have a flying speed of about 50 meters per second. And while we still can see, or while we still can discriminate leaf and soils, or plant material and soil in the first image here, back here, it's very hard to distinguish them. So you need to think about these things as well, because this also will compromise your trait extraction in the end if you don't take this into account. While these two first things are still kind of, I think, on each one's mind, more or less, then there's another thing which is not so intuitive, and you might not think about this so easily. This is the focus distance. And of course, I mean, I guess that everybody is focusing their sensing system to the right distance to the objects, and then you have sharp images. This is quite okay, but then there's another thing there, because you don't just have the focus distance, you also have a depth of field. This is kind of the range in which your objects are still in focus. And this can be very small. For instance, if you fly very, very low to get a very, very high resolution image, then your depth of field is also very small. So you might just see the top of the canopy in sharp, but then the bottom of the canopy, you will not see sharp anymore. And this, of course, then also will influence the data which you have. Or another example from forestry, for instance. So if you have very high trees and you focus on the tree crowns, then yeah, you will have a problem, because on the bottom, so on the surface, also objects will not be in focus. So this is also something you need to take into account. Still, I think most people will think about these things, but then there's another aspect. So when we reviewed the literature, we saw that most people are not reporting on these issues, and this makes it very hard to judge if, like, the quality of the data is good or not. So we found that very, very few publications are actually stating these quality indicators. And of course, this can have two reasons. So first, well, people did not think of it. Or second, it's very hard to state, because first you need to calculate them, and then you can report on them. So our approach to this was that a PhD student in our group, so Lukas Roth, has developed a very nice flight planning tool, which basically helps you in distinguishing and estimating which parameters you need for your specific objective in your flight campaign. So down here you can see a screenshot of this flight planning tool, and I have already preloaded one of our sensors. And but everybody can enter their own sensing system, save these settings, and then also load these settings if needed. So here on the left you can see the sensor parameters, so the number of pixels, the maximum shutter speed, the film speed, the maximum triggering frequency, and also the maximum flight duration of your sensing system. But also down here you can see that you can enter some lens parameters. Then in the next tab, you can set your imaging parameters. So you can set the flight height or the wanted ground sampling distance. And depending on the settings which you have, or the parameters which you have chosen for the camera, then it will calculate the right flight height. And also you can set the shutter speed that you want to use down here. Then the next tab will give you information, or will allow you to generate a GCP, so a ground control point settings, which are, so the ground control points are used for georeferencing later. Then you can set how many ground control points you want to have in every image, and also in which pattern you want to place these ground control points. And the software then will give you a pattern and a plan for your field, and then you can just put the ground control points according to this pattern, and you will meet the requirements which you have set here. Then you can also define how large your mapping area is and how much overlap you want to have. And also down here, very important, you can set the motion blur, which you want to have. And then in the end, you can, from all these settings you have put in there, you can generate a mission briefing report, which will exactly tell you how high you need to fly, how fast you need to fly, how many flight times you will need, how many flights you will need, how much flight time it will take, and so on. And then you can save this and save also waypoints for your UAV platform, which you then can later on fly. Or you can go back, adapt some settings, and see how they influence your flight mission in the end. So you can think of, okay, maybe I can go a bit lower, so I have a higher ground sampling distance, and then you can see, okay, does this still fit my flight time for my platform? So I think this tool is very, very useful for flight planning, and even before you buy your equipment, you can enter the parameters of your equipment here, and then just test, does this really fit to my objectives I have in my flight campaign? So as you see, the tool is available online, and everybody can use it. It's free. The web site is here on the top, but also if you Google PhenoFly planning tool, you should find it. And also we just got the publication on this accepted in plant methods. So yeah, as soon as I manage to do the final proofs, it will also be available online there. So coming to my next point, on sensors you can apply on your UAV system for phenotyping. And we have been really lucky, because if you look back on the last 10 years, there's really been a tremendous increase of sensing systems which we now have available. So this slides just to show you some of the sensing systems which have appeared on the market and which we now can buy off the shelf. And one thing I really like, that also very professional sensor manufacturers that previously just have built sensors for airborne or even spaceborne platforms have now started to enter the market. And nowadays we have like really, really high quality sensing system available that gives us really reliable information. And I think this is now a game changer, because, well, if I talk to my colleagues in the remote sensing community, sometimes they say, yeah, drones, that's nice, and we can do some nice things, but yeah, they are more or less toys. But I argue now, since we have these very, very high quality sensing systems available, and also the very reliable platforms, we can now get reliable data out of this. And the drones have transformed from toys to real sensing platforms. If you want to kind of classify these different sensing systems, then we have basically three categories. So the first category of spectral sensing systems are point sensing systems. They, within every integration or within every kind of capture, they take one signature of spectral information from one point in space. And then if we want to know where this information comes from, we need orientation and position information. And this is mainly done by an IMU, so an initial movement unit, and GNSS, so for instance GCP information, to then estimate where our platform was and where it was pointing at. But also we can use machine vision and the structure from motion approach and ground control points to position our information. So basically take an image at the same instant as you take the spectral signature, and then you also know where in space you have taken the information from. And it's very important to think of these things, because of course, if you need to carry an IMU and a GNSS system with you all the time, then also this increases the payload capacity, which is used by your sensing system. The next option you have are push-broom sensing systems. And these have been out there for a very long time, so I guess you are all familiar with this. So basically, these push-broom sensing systems record one line of spectral information within each integration. And then, if you want to generate a two-dimensional hyperspectral scene, you need to stitch these lines together to make a 2D representation of your landscape or your phenotyping experiment. And again, you need position and orientation information for this. So again, you need either an IMU and GNSS on your sensing system, or similar to before, you can also use machine vision, structure of motion and GCP combination to estimate the position and orientation of each image line to then stitch them together. The last type of imagers which have become very popular recently are 2D imagers. These 2D imagers record a two-dimensional image of spectral information within each integration. And this is very nice because here we already have some spatial information already in our data, because basically it's an image. And so then if the platform is moving, we get many overlapping images. And this has become so popular because we can now exploit this 2D spatial information which is in our image to put these images together. So this slide basically shows the process. So we have multiple overlapping images, and these could be RGB images, but also 2D spectral images. Then we use the structure from motion approach, which first estimates features, use a feature detection in the image, and then searches for corresponding features within all these images. And based on this, the structure from motion approach can reconstruct the relative position and orientation of these images. And then we can put the images together. But also, and more importantly, we get the 3D geometry within our scene. Additionally to the stitched scene, so the autorectification spectral scene. And this is quite nice because now, if you use 2D spectral images, we can retrieve 3D and spectral information at the same time, which allows us to exploit both of these types of information for our analysis, and can see how our plants grow and how they kind of reflect our light. And well, basically, if you combine the 3D information and the spectral information, you get something that we call a spectral digital surface model, which is a representation of the surface in 3D space, linked with spectral information emitted and reflected by the objects covered by the surface. So if you now look at the figure you can see on your slide, then you have an example of such spectral digital surface model. In gray, you can see a visualization of the height, so the digital surface. And then basically what you see here is a phenotyping experiment where you have experimental plots, and then you have rows in between of bare soil. And you can clearly see that, of course, the plants are much higher from the 3D information. And the color, which you can see here, is a representation of the spectral information. And it's represented at the red edge inflection point, which basically tells you something about the chlorophyll level of the plants. So then here you can see that the different plots have different heights. So the different varieties have different heights, but also the different varieties differ in their chlorophyll level. And all of this you got from the same sensing system. So then you can do very nice things with this information. So you can use the 3D information to track the plant height growth, for instance. So this is quite now popular concept where you generate a digital surface model at different times during the growing season, and then you subtract the height from one point in time from the other, and you can see how much the plants have grown in between these two time points. And if you do this during the whole season, you get a very, very nice impression on what has happened with the plant, which plants have grown faster, which plants have grown slower, when have the plants grown, and so on. but also we can track the biochemical traits with the spectral information. So this you can see down here. Here's the spectral information of the same phenotyping experiment for 3 different times. And what you can clearly see here is that the different varieties, so here you have the individual plots of the varieties again, but the different varieties have different chlorophyll levels. But also you can see that here in the last time point, that the spectral information tells you that something has happened here with some of the plots. And this is due to lodging, where the plants fell over due to a wind event. So then you can use this spectral information also to estimate the biochemical traits, for instance. And this is all very nice because now we have all this information available and we can run our analysis procedures on this and really learn something about how our plants grow. But sometimes we also need to take a closer look because, as I said before, I think we really need to understand what we have in our data to then judge and estimate what we really see as the traits which we have extracted. So if we take a closer look to this spectral information, we can see that, yes, the different varieties are differing in the chlorophyll level, but also we have some strange artifacts within our spectral information, which you can see here. And to kind of think of what we see here, we need to go back a bit and think about like the theory behind what we are doing here. And this is my next example when I come to and we now go to data processing. So the example is imaging spectroscopy with 2D imagers. So as I said before, when we use the structure for motion approach, we capture many overlapping images and put them together. This is shown here down in this figure. So here we have one image, here we have another image, and here on the ground we have our area of interest which we want to characterize. So this could be our plot, for instance. But as we can see here, that this plot and this area of interest is captured by multiple images. And now the question is, where do we take our data from? Do we take it from this image? Or do we take it from this image? Or do we combine the data which we have captured from all the images which are available? This is a very relevant question because this actually influences the data we have in the end, in our data product. And this is shown here. So up to the left, you can see, again, one of the experimental plots, which is overlaid by spectral information. And on the right, you can see the spectral information for this plot extracted from one of the images. Now, if we go to another image, which is captured from a different angle, then we also get different spectral information, although we have captured exactly the same area on the ground. But as I said, the image is captured from a slightly different angle. So here we have captured the same area on the ground, but we have different spectral types or different intensities of spectral reflectance. And if we now extract the spectral reflectance from all the images which we have available and plot these, then we can see that the spectral reflectance is significantly changing just because of that the images are taken from different points, and the viewing geometry on the same area on the ground is different from all these images. And now the question is, of course, how do we deal with this? For this, I first, again, show you the different example or different options we have available in our software packages to extract the data. So first of all, of course, we can just take one single image and extract just the information from this one single image. Then the next thing is we can combine the information from the different images, and then we get something like this. So down here you can see the same plot again, but if you look very closely, then you can see some lines which are going through this plot where the spectral information or the apparent spectral information is changing. So we can see one kind of block here, you can see another block here, and another block here. And this is because at these places here, we have taken information from different images. So this is one image, this is another image, and this is the third image. And this is one mode which is called blending mode disabled, for instance, in Agisoft Photoscan. Now, we also could just take all the information which we have available and average all of this information. And this is what you see down here. So here we have taken all the images which we have, averaged the information for each pixel, and this is what the result looks like. And now the question is, okay, does this really matter? Is what we see here also really influencing the spectral signature which we are extracting from our plot area? And to answer this, we need to understand what we are seeing here. So, and this now, a short warning, now this becomes a bit more technical, but yeah, later on I will kind of open up this technical, ease this technical thing again. So, if we have a single image, then and extract the information from just this single image, then we just use a very, very tiny part of the whole image. And if we just use a very, very tiny part of our image, and it's very close to the nadir view, then we also just have a very, very narrow viewing geometry. So basically we stare directly into the canopy from above in a nadir viewing geometry. Now, if you use the information from a couple of pictures, then we use more of our kind of possible area in the image. And this you can see here. So here is the information which we have taken, which is from one image. Here's the next image, here's the third image. And then also you can see that since we are using more of the whole image, then we also have bigger variety of viewing geometries that we are using to characterize our plot. So this you can see down here. So instead of just looking directly nadir into the canopy, we also look slightly off nadir in oblique viewing geometry. And now if we take all images which we have available and average this information, then the plot is captured by many, many more viewing geometries, as you can see here. So more of the whole image in the end is used. And also we have a larger variety of viewing geometries. And now if we think about it, this makes a difference. Because if we look very nadir into the canopy, we'll see a much higher proportion of soil in our data than if you look from the side. So this you can try yourself if you go outside and you walk along like a cereal canopy and you stand right next to it, look inside from nadir. It will look different than if you stand some meters away and look at it from an oblique viewing angle. And this also happens here in our data product, depending on how we process our data. Now, what we often do is that we compare our data which we have captured by our drone, or also it's the same thing for satellites and airplanes. We compare this to data which we have captured with this field spectrometer. And the field spectrometer captures field spectrometer typically has a field of view of about 25 degrees, and the information which it's capturing is averaged over all of these viewing geometries of 25 degrees. So this is shown down here. So we have a much higher variety of viewing angles, which is averaged. And now if you look at this situation, I think it's pretty clear that we kind of see different things in our canopy, although we look at the same spot. To describe this phenomenon, we have come up with this term of specific field of view, which is a composition of pixels, but also together with the angular properties within a scene used to characterize a specific area on the ground. So basically what I'm saying here is that it's not enough to know where your data is coming from in the spatial domain, but also you need to think of in which viewing geometry your data is captured. Okay, now let's look at the difference between these different processing schemes. Here again, you can see the different data products on the left. and the color around the resulting data product depreciates also the spectral signature, which you can see in the middle. And by the first look, we also can see that there are differences, for instance, here in the near-infrared region. But if we take a closer look and look at the relation of the different data products, or the relative difference of the spectral signature extracted from the different data products, we can see that we have huge differences between these different, differently processed data products, of up to 10% if you look at the red edge region and the red region. So basically, if we compare the ASD measurements to the single image, then we have about almost about 20% offset in the red and red edge region. If we compare the blending mode average, which is shown in red, to the single image, then we have about 10%. And while the blending mode disabled is more comparable to the single image, which is shown here. So yeah, some 3–4% difference. And now the question, of course, again is, yeah, we have the differences in the spectral signatures, but now if we use our retrieval methods, do they normalize for this effect? And the short answer is unfortunately not. So normally we use vegetation indices to kind of to normalize illumination differences and so on, and also to, of course, retrieve our parameters which we want to have. And what you see here are the results for 4 different vegetation indices. On the x-axis, you can see the result for which you get from a field spectrometer. So if you calculate the vegetation indice from the field spectrometer data, and on the y-axis, you can see the result if you calculate the vegetation indices for the different experimental plots from the imaging data from the drone. And what you can clearly see here is that you have an offset from the 1-to-1 line, which means basically you also have an offset of your kind of retrieval of your retrievals, depending on which sensing system you have used. But what you also can see, and this is preached by the color here, that these differences change from one growing period to another. So the different colors stand for the days after sowing, different days after sowing. And you can see that for some vegetation indices, it's pretty constant throughout the whole growing season. But for some others, there are differences. And of course, this makes sense because, well, during plant growth, also the structure of the plants change. So the influence of the structure and the viewing geometry will also change. So what is the take-home message here from this second example? The take-home message here is that you need to think of what you are doing during your data processing. And then you need to think of what you are comparing to what. And as I showed you, depending on how you process your data, you will have different data properties, and you will measure different reflectance factors. This is the remote sensing term for this. And you need to take this into account if you want to interpret your data in the end. And then, of course, if you want to compare data from one field site to another, you should also process it in the same way, because only then you get comparable results. With this, I come to the last topic of my talk, which is on plant trait extraction. And before I go to this, I would like to introduce you our field site here at the plant research station of ETH and Aasen. And for this, I first show you what kind of equipment we have here. So our kind of backbone of our research is the field phenotyping platform. It's a very nice platform. Yeah, it's a very nice platform, and it's similar to these camera systems which you know from the soccer stadiums. So we have 4 poles around about 1 hectare field, and on these poles, we have a cable-suspended camera system which can freely move over the whole field. And we have equipped this with a lot of sensors. So we have a LiDAR system on there. We have a hyperspectral system on there. We have a thermal camera on there and a high-resolution RGB camera system on there. And this allows us from a distance of about 2 to 3 meters above the canopy to really look at individual leaves and what is happening there. So there we are on the leaf plant to plot scale level. Then we use our multi-rotor UAVs, which are equipped with hyperspectral, thermal, and RGB sensors. And these we use from the plot to field scale. So for everything which is around or below, let's say, 2 hectares. Then last but not least, we also have our fixed wing UAVs, which we have equipped with multispectral and RGB sensors. And we use these to fly from the plot to the field to regional scale. And they are like within every flight, they can capture about 50 hectares of area. Then most of our experiments are carried out at the plant research station at the ETH in Eschkon-Lindau. And to kind of introduce this field site to you, we have produced a small movie which I will just show you. And I assume that it will not be very fluent through the join me, but I will stop at important positions of the movie. So down here you can see the 1 hectare field with the poles of the FIP. So we have a typical crop rotation for Switzerland with maize, wheat, soybean, buckwheat, and some grassland over here. And down here, I will zoom to it. Here, you can see our FIP, which is moving across the field and is scanning the individual plots. So our wheat experimental plots have about 350 different varieties of wheat. Our soybean experiment has about 60 different varieties in, I think, 4 repetitions, and the wheat have 2 repetitions. And up here you can see our drone is flying. And this is a key concept because we want to kind of compare what we see on the different levels from the drone and from the FIP. So we mostly apply all the different systems at the same time. What you can also see is that we have equipped our field site with fixed ground control points. So down here on the bottom of the pole, you can see that we have GCP, we have one here, we have one here, and all over the field we have distributed them. And they are staying there. And this is very important because only this allows us to then put data from captured at different times together. Then back here, you can see that we have also laid out radiometric reference targets. These radiometric reference targets are used to then normalize the spectral information from one time to the other. And we always use 5 targets, and then we use 3 targets. We use 3 targets to calibrate our data with the empirical line method. And then we have 2 targets left to do quality assurance. So overall, we have a quite nice setup, I have to say here. But I would definitely recommend you, if you do such experiments, to also put ground control points at the spot and also these radiometric reference targets, because this will speed up your research and your experiments much more. Because, I mean, flying takes about 10 to 15 minutes for the whole area, but distributing ground control points and radiometric control points and so on, this takes half an hour. So basically, you can cut down the time you need a lot if you just equip your field site with the appropriate things. Okay, coming back to the slides. What do we then get out of this? So if we do one flight, then we get a nice automosaic, for instance, of RGB data with a ground resolution of down to 3 millimeters. So basically, we can even see individual plant organs in this data. Also, what we can clearly see already on this RGB image is that we have a nice differences between the different wheat varieties, for instance. And since we are flying very often, so we fly about 1 to 3 times a week during the growing season, we can then also have visualize and explore what is happening from one point in time to the other. So down here you can see the development of wheat of one plot through the time. So in the beginning you could see that there are clear differences in the early vigor between the different varieties. And also in the end here you can see that also the senescence process is strongly differing between the different genotypes. So we'll just play it once more. And now, of course, you could ask yourself, well, now we have very, very high resolution data. So we have captured it very often. We have a very high spatial resolution. So what can you now do with this data? And with this, I would like to come to my last example of extracting leaf area index using viewing geometry effects. Well, a couple of minutes ago, I was introducing that depending on which viewing geometry you have and basically where your image is captured has an impact on the data you get. And this can be a backdraw and a challenge, but it can also be used. So for instance here, this is shown in this figure. So down here you have your area of interest, so your plot basically, and up here you have the different images which capture this one plot. So if you look at the plot in a very nadir geometry, then it would look something like this, which you can see here on the right. And of course, the right is just a model. Now, if you capture the same plot from a different position, so you have a different viewing geometry, your plants will look different, although they are, of course, the same plants. And if you think about it, depending on which viewing angle you use, you will see different things. So, for instance, a soybean plant would look like this if you would look at it from an oblique viewing geometry. If you would look at it from a nadir viewing geometry, you would just see this. And if you would look from it from the side, you just see this. So basically, depending on from which angle you look at it, you get different information. And this is very nice because, well, we have a lot of viewing geometries available. And if we explore and exploit this fact, then we can make our data extraction and our trait estimation much more robust. So what we have done here is that we have taken all the images which we had available, estimated the camera position and orientation for each image, georeferenced the image, extracted each plot from each image which was available, segmented plant from soil background from it, and then calculated the green cover. Here you can see an example of this. So in the first column, this plot is captured by a nadir viewing geometry. As you can see the original image here, the segmented image down here, and the plant and the soil kind of extraction and mask. Then in the next column, you can see how the same plot and the same area looks if you look at it from a 20-degree viewing geometry. And the last figure here shows how much of the area was estimated plant. So from this nadir viewing geometry, we have about 60% which is estimated to be a plant. If we are at a viewing geometry of 20 degrees, then we already have 68% green cover. And now if you go to a viewing geometry of 30%, then we already have a green cover estimation of about 72%. And now we can use this together with a plant growth model and combine it. So the information, the simulations of a plant growth model and our extracted green cover from these different viewing geometries to estimate the leaf area index. And then we get something like this. So here we have the leaf area index, which is estimated by a Lycor system down here to the right. And to the left, we have the leaf area index, which is calculated from this method, which I just introduced. And what we found is that, well, if you use your Lycor system, so basically you go to every plot, apply your Lycor system a couple of times, and then you retrieve the leaf area index from it. This will take a very, very long time because, I mean, if you want to screen 700 plots, this is very laborious. While the drone flies above the field and is done in 10 minutes. And if we now compare the data, then from an R-square perspective and also from the intercept and the slope, we get very, very similar results from our drone image and drone approach than we get from this very laborious ground approach. And I think this is a very nice example of how you can use drone images and explore this kind of multi-viewing geometry effect thing to get a very reliable extraction of a plant trait. So in this case, a leaf area index. So with this, I come to the conclusions which I have from my talk. So I really believe that UAVs are the perfect tool for field phenotyping. Yes, large infrastructures such as the FIP, they are much more reliable and can look at data much more precise, and I like it a lot. But if you want to go to different environments, then drones are really a cool tool. They're quite flexible also, and they deliver very high temporal and spatial resolution, which we can use for our phenotyping applications. And as I also showed you in this talk, if you know what you do, they can provide you with reliable data. And I think this is a very crucial part because this has not been the case for all the times because the sensing systems and the drone systems were not like so mature as they are today. But of course, you need to do what you are doing. So with the drones, it is your responsibility to get reliable data. So if I think back to like classical remote sensing, then you have a few data providers which just give you the information. And they take care of all the pre-processing and geometric processing and so on. But now, if you use drones, it's your own responsibility, and you need to think of how to get there to get reliable data. And this is just possible if you know your sensing system and also you know your flight parameters. And this is why I think tools like the PhenoFlight planning tool are so important, because there you can really generate and think of what you actually are capturing, and you can also generate quality parameters like estimation of motion blur, for instance, from such tools, and these give you a feeling of what you're actually doing and what your data, what you are capable of if you have the data available. So, bottom line of this is you need to think of what you want to measure and what you are really measuring, because these could be two different things. But if you take this into account, and if you take a couple of minutes to think about it, I think it's really possible to go this way. And I think really that drones will kind of change how we do field phenotyping now and also in the future. With this, I'm at the end of my talk. And I would like to say thank you to a couple of people from my current group here. So first of all, I would like to thank Professor Achim Walter, from the professor here in our crop science group, which has made all of this possible. Then also, of course, I would like to thank the PhD students, which currently also work in the PhenoFlight team. So Francesco, which is in a dig-in project, and Lukas Roth, which is working in a trade spots project, and Kirina Merz, which is working in an InnoFarm project. Then I also would like to thank Norbert Kirchgessner, who is the master of our FIP system, for his constant support. And then also Frank Liebisch for his constant support with the PhenoFly, and also for being like a senior researcher in our team and gives to give advices. And also I would like to thank Andreas Hund, who is kind of the master for the breeding questions in our group. So with this, I'm finished. If you would like to stay updated about what we are doing, then follow us on Twitter or visit our website or just drop me an email. And with this, I also would like to thank you for your attention. So close to Christmas, it's nice that you take your time and took the time and listened to what I had to say. And if you have questions, I'm happy to answer them. Thanks a lot.
Okay, thank you very much, Helge, and thank you as well for doing it close to Christmas. It's the same for you as it is for your audience. I've been watching the chat box and looking to see if there are any questions already. The only one I can see at the moment is about specific field of view, and there are a couple of people chatting about whether it's a function of image overlap, whether having an 80% versus a 60% image overlap will influence the analysis and what the effect of that is. I wonder if you can say anything about that.
Yeah, sure. So the specific field of view kind of is a function of a lot of things. So of course the overlap influences it, because if you have a higher overlap, you will have more viewing geometries. So basically, I will go back to one of the slides. So if you look at this slide, basically if you would have a higher image overlap, then basically imaging positions would be closer to each other, and you would see the plot from more angles. And of course this would have an influence, but what has a bigger influence is that your flying pattern is kind of regular. So for instance, if you have a 60% overlap in one direction and an 80% overlap in another direction, which is very often done in remote sensing, then you will have like a bias in your viewing geometries towards like either like a long track or a cross track flying direction. And this will then kind of unbalance your viewing geometries if you average all the information which you have available. And then, of course, it also depends on the field of view of your sensing system. So if you have a very wide field of view, as it is normal for RGB cameras, then you will have a lot of viewing geometries which can be mixed if you average all the data you have available. While if you have just a very narrow field of view, then of course also the viewing geometries are more restricted. So these are basically the three parameters you need to think of if you want to know kind of this specific field of view thing.
Okay, thank you. So there's a question from W, which is where or how can we get a reliable radiometric calibration panel?
Yeah, this is actually a very good question because, well, this is just the basics you need, but it's not so easy to get a reliable radiometric calibration panels. So there are a couple of options available. So the first one is you go to a manufacturer of a spectrometer, and so to Sphere or to a reseller of spectrometers, so for instance to Sphere Optics, and you buy a barium sulfate panel or a couple of panels. These are very reliable, but they're extremely expensive. So a small panel already costs in a magnitude of €1,000. So this is not really feasible if you want to have multiple panels. A better idea, and this is what we have done, is there are some paintings around which have quite Lambertian behavior. So the color we have used is the Nextel color, and you can buy it online. And there are some car painters which can kind of put this color on panels, and you could then kind of let these panels manufacture for your own use. And these come much cheaper. So I'm not sure how much we pay for each panel, but it's rather in the magnitude of 100 to 150 euro. Then another thing which I know of is there are some sailing cloth around, which also have a quite good Lambertian features. But I cannot recall the name of it. But these are also, of course, then much cheaper than like the really professional panels.
Okay. So there's another question just popped up that I'm not sure I fully understand, but maybe that's my unfamiliarity with remote sensing. What is the reference of the color?
What is the reference of the color?
So I think that's the radiometric calibration panel, isn't it? That's telling you.
Yeah. So basically, I'm also not 100% sure where this question is aiming at. But yes, the reference of the color, what we do to kind of know what reflectance our reference targets have, we measure them with an ASD field spectrometer. This is basically our standard. And then we resample or convolve the bands of the field spectrometer to the bands of our, for instance, spectrometer or imaging spectrometer on a drone. And then we apply the empirical line calibration method to kind of then calibrate our drone images. But of course, the prerequisite to this is that you also have already a calibrated system on the drone. So this just helps you to do kind of atmospheric correction, while the other artifacts in your images, so the vignetting, for instance, of your lens system, these you need to characterize before and normalize for this before.
Okay, thank you. Yeah, I think that does answer the question. I think I understand the question now. It's me being silly. So we have one more. If you limit the specific field of view, can you extract the plant height from your DSM data and generate a reliable plant height time series?
So, good question. Well, I would say yes, if you then have a much higher, basically in the end you need a high overlap of the images to stitch these together and to then reconstruct it 3D geometry to get your digital surface model. But I'm not sure if I would do this, because then of course you also need to fly much slower to get the overlap, or you need a much faster camera system. So what I rather would like to do is just keep it this way, but then during your processing implement kind of a tracing method, so you know what kind of how big the specific field of view for the area of interest was, and then you can basically trace this and estimate this.
Okay, so there are a couple more. So how do you deal with cloudy conditions? Says BS. I think you don't have so many sunny days in Switzerland.
Haha. Well, you should have been here this summer. It was like just perfect days all the time. No, of course not. But yes, it's a very relevant question, and I didn't address this in this talk. But so what I think, it's generally not such a big problem if you have clouds, because more or less recently there was a method published which is called radiometric block adjustment. This basically works like this, or like such that you search for radiometric type points within your images. So basically one area which is visible in two images, and then you estimate the reflectance from this image. And then you basically normalize the next image based on these areas which are visible in two images. And then you can normalize the differences in between, like the illumination differences between two different images. But of course, this just works as long as you have more or less constant conditions. And by constant, I mean either sunlit or mostly sunlit, so mostly direct illumination conditions, or just diffuse illumination conditions, such as if you have cloud cover. As soon as you kind of move from direct illumination to diffuse illumination, you will have a problem, because then you have other effects like shading within the canopy, which will change. And this is, in my opinion, almost impossible to normalize for. So it's not such a problem that if you have clouds, that it becomes a bit lighter or a bit darker. This you can normalize. But if you have really changes from direct to diffuse or the other way around, then it becomes very hard. And I have to admit, I don't have any solution for this case.
Okay, thank you. So perhaps one last one, and then we'll let you go. So do you have a solution to evaluate the quality of point cloud data? Well, I suppose we have two questions popped up at the same time, both about characterizing the quality of various things. So basically, how do you evaluate the quality of the point cloud data that you get from structure from motion? Yeah. Do you characterize the radiometric stability of your cameras in the lab?
Yeah. Okay, I will start with the second one because this is the more straightforward. Yes, I do. So what we do is we place the camera in the lab, and then we measure it. We increase the ambient temperature, we decrease the ambient temperature, and see basically how the camera reacts to this. We record constant, like of the same target, we record constant data when the camera is heating up. And this also will give us some information how stable the radiometric calibration is during different temperatures of the camera. So yeah, we do this. And I think also, yeah, you need to do this if you really want to have reliable information. Then for the first question regarding the quality of the point clouds by structural motion, yeah, very, yeah, let's say interesting and challenging because it's hard to evaluate. I mean, it's easier to evaluate the plant height, which you get from a DSM. You could compare it to either ruler measurements or what we have done, or what Nora Tilly actually has done a lot, like from the University of Cologne. She has compared this to laser scanning data. And for the height, this is quite straightforward. And we can see that there are some small offsets which result from how the structure from motion process basically works and which features you see of the plants. But if you really want them to go to the point clouds, I think this is much more challenging because, well, again, there the viewing geometry has a huge impact. So basically, of course, again, if you don't have so high overlap, then it's not so likely that you will have rays which will meet lower inside the canopy. So we will miss out points from lower inside of the canopy, and you have a tendency to just capture the top canopy. While if you have a very, very dense flying pattern, you have a higher probability to also see more into the canopy, and this will influence it. So it's very hard kind of to estimate the quality of the structure from motion point cloud. The best way, I guess, would be to just compare it to laser scanning data again. But then you need a system which is right above the canopy. And we are currently working on this, but I can tell you already now, it's not so straightforward.
It is a very difficult problem, isn't it? Because getting ground truth of almost any measurements of plants is difficult given their complexity. The thing that we did with single plants at Nottingham, which you can't obviously do in a field, is when we were reconstructing 3D models from multiple views of individual plants, we X-rayed the plants beforehand and used the X-ray data. So then we had still problems that the plant would move and deform between two operations. But we had something that at least captured every surface of the plant. Yeah? But it's a very difficult problem.
Yeah.
Okay. I think we should perhaps call it a day there and let Helge go and prepare for Christmas. Thank you very much. It's been a very interesting talk, and you can the way you can tell that is the number of questions that you generated and the size of the audience. So thank you very much. Thank you everybody as well for logging in and listening. The recording will be put on the IPPN Working Group website as soon as it's ready. It has to be processed, but it will be up there as soon as it is. So thank you once again, and have a good Christmas.
Yeah, thanks a lot to everyone, and thanks to you, Tony.
Okay, you're welcome.